Let's see about the cells of inflammation. I'm going to talk to you about neutrophils, macrophages, and all the other cells of inflammation in a very interesting way. Stay tuned. So, what is neutrophils? Have you ever wondered how your white blood cells actually kill the bacteria? They don't swallow them whole like Pac-Man. They unleash a chemical storm. Let's follow oxygen as it transforms from a harmless molecule into superoxide, hydrogen peroxide, and finally hypochlorous acid. The very same chemical found in the bleach is right inside our body. So let's see how it does. Neutrophils are the first responders at the sites of inflammation, rapidly recruited within the first 24 to 48 hours of injury or the infection. So how does it do? What does it do when it comes across the microbe? They are highly phagocytic cells. They will kill and they will engulf the microorganisms. Following that, they will use the respiratory burst, that is uses the NADPH oxidase to generate H2O2 and myeloperoxidase to produce bleach. How do they do that? The molecular oxygen which is present inside the cell is converted by NADPH oxidase to superoxide radical O2 minus. This is the superoxide radical. Superoxide dismutase is the enzyme that converts superoxide radical to hydrogen peroxide, that is H2O2. The myeloperoxidase combines the chlorine to hydrogen peroxide, forming hypochlorous acid or the bleach. So this is the respiratory burst pathway that happens inside the neutrophils. So another fact is, how does the neutrophils reach that particular site? It is by means of chemotaxis factors. They are C5a, interleukin 8, kallikrein, platelet activating factor, as well as the microbe antigens. So all these are chemotaxis. In other words, they will be attracting the neutrophils, or they will be calling the neutrophils to that site. Neutrophil clinical correlations. Normally 50 to 60% of the WBCs are neutrophils. When there is a more than 60% of the WBCs are neutrophils, it indicates neutrophilic shift, suggesting bacterial infections. Now what about bands? What are bands? Bands are immature neutrophils that are increased in bacterial infections. So they are the immediate precursors to the fully mature segmented neutrophils. The mature segmented neutrophils will have the classic multi-lobed nucleus. But bands, they have a C-shaped, horseshoe shaped, or even U-shaped nucleus. So that's why it is called bands, because there are no thin filament connecting the lobes. So it's called banded. Bands are immature neutrophils. When the total WBC is more than 30,000. Normally it will be 4 to 11,000. When the total WBC is more than 30,000, it can be either leukemia or it can be due to reactive granulocytosis. What is the other name for reactive granulocytosis? That is leukemoid reaction, because it looks like leukemia. Oid means it looks like leukemia, but it represents massive neutrophil release from bone marrow reserve. Therefore, it is not leukemia, but it is a reactive granulocytosis. It is reacting to the virus or the pathogen entry by increasing its neutrophil count. So we should not be alarmed that it is a leukemia, but instead we have to identify it as a leukemoid reaction. What does the neutropenia causes? The neutropenia, that is reduction of the neutrophils, can be seen following chemotherapy, following radiotherapy. Sometimes certain viruses will cause aplastic anemia, bone marrow suppression, therefore resulting in decreased neutrophils. That is the parvovirus B19. In addition to it, certain drugs like clozapine, methotrexate, carbimazepine, methimazole, propylthiouracil can also cause neutropenia. What is accumulation of pus? What is pus per se? Pus is accumulation of dead neutrophils, cellular debris, pathogens, as well as the tissue fluids. Pus is something which is good because it has entire thing and it is trying to eliminate the body. Have you ever wondered what happens in your body when an infection sticks around for weeks instead of days? The neutrophils leave and the macrophages step in. They are not just to eat up the invaders, but to coordinate the immune response, like how a general would behave on a battlefield. Let's explore how these chronic inflammation specialists, macrophages, work, from calling in T cells to unleashing the cytokine storms. The macrophages predominate after 48 hours. This is the exact timeline, replacing the neutrophils in chronic inflammation. They will serve as antigen presenting cells, thereby exposing the phagocytosed antigens to CD4 T cells. In chronic PID, they will be presenting with a scarred fallopian tube. The macrophages are the ones which will predominate on the biopsy of the scarred fallopian tube, because in the chronic inflammation is caused by macrophages. There are some cytokines which are produced by the macrophages. They are tumor necrosis factor alpha. So all these help to clear the microbes. How? The tumor necrosis factor alpha will increase the vascular permeability and will cause swelling. Interleukin 1, interleukin 6. Interleukin 1 will cause fever, and interleukin 6, it is an acute phase protein. So interleukin 12 will cause a stimulation of CD4. So in this way, by increasing the body temperature, by increasing the permeability, by causing the swelling, that pathogen is finally cleared from the body. In addition, macrophages play a very important role in septic shock. How? There are two types of shock. I have a detailed presentation about this. If you are more interested to know about this, you can browse my immunology playlist. Macrophages in septic shock. They cause endotoxin induced shock and superantigen induced shock. In endotoxin induced shock, the lipid A of the lipopolysaccharide, which is a part of the endotoxin, that is released by the gram-negative bacteria. That lipid A will bind to CD14, or the toll-like receptor of the macrophages, thereby causing degranulation of the macrophages. Therefore, the TNF alpha will be released, which is increasing the vascular permeability and vasodilatation. And interleukin 1 will be released, which will be causing fever. This is about the shock in endotoxic shock. What about the superantigen induced shock, which is seen in Staphylococcus aureus or Streptococci pyogenes? Here, this superantigen, which is released by these microbes, Staph aureus and Strep pyogenes, will bridge MHC class 2 and the T cell receptor, resulting in massive cytokine release, resulting in the toxic shock syndrome. So both the pathways will finally lead to the similar clinical presentation, but macrophages play a pivotal role in them. They also have formation in granulomas. What are granulomas? Granulomas, the name looks different, but they are also activated macrophages. We do have another name for activated macrophages, that are the epithelioid cells or histiocytes. The activated macrophages will fuse to form multinucleated collections. Why do they fuse? They fuse in an attempt to digest the foreign antigens. So this is what a granuloma is. Where do we see granuloma? This granuloma can be caseating, that is, it will look cheese-like, or it may not be caseating, may not look cheese-like, that is non-caseating. So caseating is seen in tuberculosis, cat scratch disease, fungal infections, while non-caseating is seen in sarcoidosis and Crohn's. There are few more roles for macrophages. Atherosclerosis. The macrophage will phagocytose the oxidized lipid particles in an attempt to eliminate the lipid particles from the body. But they don't really get to eliminate it, and they finally contribute to the plaque formation in atherosclerosis. They also promote the granuloma of granulation tissue and fibrosis. The macrophages which are present in the liver is known as Kupffer cells, while in the central nervous system is known as microglia, while in the lungs it is known as alveolar macrophages. So we have seen a lot about macrophages. Let's move on to our next factor, that is the transforming growth factor beta. Transforming growth factor beta. So this transforming growth factor beta will stimulate the proliferation and activation of fibroblasts and myofibroblasts. Before I go into detail about fibroblast and myofibroblast, I would love to tell you an analogy over here. Imagine a construction site. Fibroblasts are like the bricklayers and cement workers. They bring in the raw material. Over here it is the collagen or the extracellular matrix, and they build the walls and foundation of the repair site. Without them there is no structure. What do myofibroblasts do? They are like the workers who tighten the scaffolding with ropes and pulleys. They do add more bricks, that is, they add more collagen in our context. They also pull the wound edges together, contracting the site so the gap closes faster. So what do we know from this analogy? The fibroblasts and the myofibroblasts promote collagen production and are essential for tissue repair. There is a catch over here. If the myofibroblasts don't leave after the job is done, then they keep pulling and laying down excess cement, leading to an overbuilt or a rigid structure, finally causing fibrosis and scarring. So how will this knowledge be asked? You assembly can ask you which cytokine helps in extracellular matrix homeostasis. That is transforming growth factor beta. That is the answer. Platelet derived growth factor. As the name says, platelet derived growth factor, they will stimulate smooth muscles and endothelial cells, contributing to the new blood vessel formation. In addition, they can also recruit and activate the fibroblasts at the site of injury. Angiogenesis is by VEGF. Obviously the name vascular endothelial growth factor. The vascular endothelial growth factor is the primary stimulator for angiogenesis, promoting the formation of new blood vessels. How does this happen? Let's see. There is an initial tumor growth. So when the tumor grows, then the blood supply is constant. Blood supply is not increasing, while the cell number increases in the tumor. Therefore, we have to feed a larger number of cells over here, creating a higher oxygen demand. This will result in low PO2 because they are suffering from hypoxia because the blood vessel is the same. Therefore, the low PO2 falls due to increased cellular oxygen consumption by growing the tumor mass. And this hypoxia will stimulate the vascular endothelial growth factor. That is, it will cause the upregulation of VEGF, which will stimulate the angiogenesis to happen, thereby providing adequate blood supply to support continued tumor growth. We do have something known as fibroblast growth factor. How do we know this? How do we remember this? The fibroblast growth factor is the body's superhero. It grows blood vessels, calls fibroblasts and skin cells into action, and it even shapes the bones, powering early wound healing like a boss. What do I mean by this? So it will stimulate the rapid multiplication of fibroblasts at wound sites, thereby providing cellular foundation. Fine. Then it will promote the collagen production by the activated fibroblasts. The fibroblasts are brought there and that is providing the collagen. And it will also result in tissue remodeling. that is restructuring of the damaged tissue. When there is a problem in fibroblast growth factor receptor, when there is a mutation, it can even result in achondroplasia, that is poor skeletal development. So in short, it will stimulate fibroblast proliferation, keratinocyte and endothelial cell migration, angiogenesis, as well as skeletal development. What about epidermal growth factor? As the name says, it provides proliferation and migration of epithelial cells, which is the final step, or it is essential for re-epithelialization and wound closure. It is crucial for restoring the barrier function in wounded tissues. Therefore, preventing the infection and promoting the healing. With this, we have come to the end of this presentation. I hope you have liked it, and I tried to make it as interesting as possible. If you like my videos and if you want to get mentored by me, you are more than welcome. I mentor medical graduates and USMLE aspirants, those who are preparing for USMLE. Thank you. Have a good day.